thus far in your immunology education, I've been playing kind of fast and loose with this term antigens, kind of assuming you know what I'm talking about and going along with it. For example, I told you that the innate immune response is not antigen specific, while the adaptive immune response is antigen specific. But what exactly is an antigen? Well, technically, an antigen is any molecule that can be recognized by the immune system. But that's not really the whole story, right? Because it has to be more than that. We know that there's non-specific responses and specific responses. So how do we break this down further? So in the innate immune response, you have pattern recognition receptors and you have PAMPs, right? All right, so the PAMPs are the pathogen-associated microbial products that the pattern recognition receptors recognize. Now, technically, when a phagocyte like neutrophils binds to LPS on an E. coli bacterium, that's nonspecific. It doesn't know what it's binding to. It just knows it should eat and destroy that. With non-specific responses, it's looking for a very specific amino acid signature that always will be the same, okay? And it's always going to look the same, and therefore the receptor is always going to be able to bind exactly that signature. If it changes even a little bit, it's not going to recognize it. With the adaptive immune response, we're thinking of very specific types of antigens. So when I say the term antigen specific, I'm really only referring to T cells and to B cells. Now, I want to make one other thing pretty clear. When a substance can react with and activate the immune response, it is referred to as an immunogen. That means it is leading to an immune response. The terms antigen and immunogen are often interchangeably used. Even though all immunogens are antigens, not all antigens are immunogens. Just because we bind something and recognize it doesn't mean we're going to get a response. I liken this to just because you go on a date doesn't mean you're going to marry the person. There might be no spark. The same needs to be true for T cells and B cells. They need a spark. So let's go through some guiding principles of antigens. First off, only a limited portion of the antigen molecule is actually going to interact with the receptor. So if I have an antigen, and let's say this is its amino acid signature, okay? It has these amino acids in this order. My TCR or BCR might only recognize these 3 amino acids or the 3D conformational structure that these 3 amino acids take on. It doesn't mean that these aren't part of the antigen. They are. It's just not the part of the antigen that my epitope receptor is recognizing. And this section, the section of the antigen that my antigen receptor is recognizing, is referred to as the epitope. Another set of terms you might hear for epitope is antigenic determinant, but I'll try to use the term epitope as it's technically more accepted. The two terms are often used interchangeably. The number of distinct epitopes within an antigen varies with the size and complexity of the molecule. A given individual really only respond to one subset of epitopes. Now, epitopes that induce an antibody response in the majority of people. So in your class of 100 and almost 50 students, if in this class of 150 students, 90 of you respond to epitope A, and another 10 of you respond to B, and the rest of you respond to C, epitope A is going to be referred to as the immunodominant epitope because this is the epitope that whatever antigen we have, the majority of people are responding to. Why is this important? Vaccines. When we are designing vaccines, we want to make sure we're designing them so that the majority of people will make a strong antibody response because that's the best way to increase the herd immunity to that epitope. Okay. Now, let's make things even more complicated. When we think about antigens and epitopes, a single antigen like this one, so I'm going to rewrite it over here. We had T, R, S, G, and A. Okay, a single antigen may have more than one epitope in it that are recognized by different TCRs and antibodies. Okay, so maybe T cell number 1 responds to this epitope, whereas T cell 2 responds to this, and the antibodies made by B cell 1 actually respond to all 5. See what I'm saying? So within this one antigen, there's actually 3 different epitopes. Think of this like a lock and key. In this example, your hand is, say, the MHC molecule that's holding up the key. Just looking around for my keys. I can never find them when I need them. So my hand is the MHC molecule. If all of the doors in the world, or at least at Rush, are considered the antigen, right? Each door lock is going to have a particular epitope that on this key, that key ring that it'll bind to. So like one will go to my office, one will go to my lab, one will go to my car. And so this is an antigen, but it has different discrete epitopes for whatever I'm trying to access. Okay? All right. So while we're talking about vaccine design, let's talk about something that's closely related to it, and that's known as haptens. Haptens are not immunogenic, but they are sometimes used to boost the response, particularly in vaccine design. Haptens are small, chemically defined molecules that are not immunogenic but can react with antibody of appropriate specificity. They're the one class of substances that are antigens but not immunogens. They are always univalent molecules, meaning that they have a single epitope. They don't have multiple epitopes. A really common hapten that's often used in vaccines is DNP, dinitrophenol. It basically acts as a partial or complete epitope when coupled to a protein carrier, and that's what's used in vaccines. The protein carrier will have its own inherent epitopes. Some antibodies might be made against the hapten carrier complex, which will be specific for the hapten instead of the pathogen, but the benefit is still that we've boosted the protein or the response to that peptide that is associated with the pathogen. Okay, now the other thing we need to keep in mind for epitopes is that both the conformation and the sequence are important. So if you have a antigen that arranges itself in beta sheets, that might be important, or in helices, that might be important. Or you might have an antigen that is strictly based on the sequence of the amino acids. My most favorite of these is OVA. Ovalbumin is actually the most feared molecule in all of research. It has to be because people study it all the time. In certain mouse strains, ovalbumin from chicken eggs is the only thing that a lot of T cells will respond to. So this is actually the amino acid signature for the antigen, okay? And there's an epitope in here that a lot of T cells will respond to. And that's just based on amino acid sequence. But there are other T cell and B cell responses that are really based on the 3D conformation. So the 3D conformation of the epitope might affect how the antigen is seen. And the sequence of amino acids may be necessary to form this conformation. So if you alter the sequence of the amino acids, now suddenly the conformation won't be the same, and the T cells and B cells might not recognize it. Why is this important? Think of viruses that mutate really quickly. So an example of this would be like HIV. HIV has glycoproteins on the surface, but HIV mutates really quickly, a lot of these glycoproteins. So if previously the structure was, a beta sheet and I had antibodies that could bind that and neutralize it, great. Now it's mutated, let's say, so that now it's a helix. Now my antibody can't bind it as effectively, and now it's no longer a neutralizing antibody. So this could be really, really important. And that also tells you that denaturing the antigenic molecule might affect your ability for the cells that you're looking at to be able to see the antigen effectively. Antigens can come from all sorts of spots on a pathogen. They can be external or they can be internal sources. So what do I mean by external versus internal? So an external source would be something that the pathogen produces. So a good example of this would be like a toxin, okay? We make antibodies to tetanus toxin. We make them too slowly to help us, but technically we do make antibodies to tetanus toxin. It can also be to internal sources. So an internal or endogenously derived antigen would be something like on the cell surface of the molecule. So for instance, some of the antigens on the surface of strep pneumo. We make specific antibodies to the different serotypes of strep pneumo, and that's part of the reason we know there are serotypes, because there's a lot of different ones. The way T cells and B cells identify antigens is through specialized receptors. These receptors are known as the B cell receptor and the T cell receptor. Okay, the T cell receptor is always attached to the surface of the T cell, while the B cell receptor can at times be secreted as an antibody, and that antibody is going to have the exact same antigen specificity as the BCR of the B cell that created it. Antibodies and the BCR can see all sorts of different extracellular antigens in any context, and that includes like carbohydrates, lipids, proteins. They can see it all. T cells are much more selective. They're like the divas of the immune response, and they really only see peptide antigens, and they have to see them in the context of MHC. So keep that in mind as you're studying the different ways that antigens can be recognized.